last time we were talking about blood agar plates. And so remember, this is an enriched media. It has whole red blood cells added to it. Because of that, it's also a differential media. It helps us differentiate organisms that are hemolytic. So last time we talked about alpha hemolysis. And remember, that's incomplete hemolysis, and the plate's going to look kind of greenish. As opposed to beta hemolysis, what happens to the red blood cells? They're completely destroyed, right? So things like strep, the causative agent Streptococcus pyogenes, does stuff like that to red blood cells. Other bacteria will grow on this media because remember, it's usually a complex media that they've added the whole red blood cells to. So other organisms will grow, but they won't cause any changes to the whole red blood cells. So we refer to those as non-hemolytic, or sometimes you'll hear the term gamma hemolytic, which I don't like. It's very, the word just skipped right out of my head. It sounds like hemolysis, right? Hemolytic sounds like hemolysis, but gamma is not lysing the red blood cells at all, right? So it's misleading. That's the word, misleading, right? It's misleading. So I usually stick with non-hemolytic, right? That's pretty straightforward. It's not lysing red blood cells. So you can see growth on these plates, but no change to the red blood cells. And here shows all three types. So alpha, you have that greenish decolorization. Beta, you have the complete clearing. And gamma, you have just the growth and no change to the red blood cells. Now, McConkey agar, which we don't use in the microbiology lab, we use one similar to it, is both a selective and differential media. Selective means what? Means we're selecting for a particular group. How is it that we can get one group to grow and others not? What do we do to the other ones? We inhibit their growth. There's something in that media that's toxic or inhibitory to the organisms we don't want growing, and thus allowing us to select for the ones we want. So in the case of McConkey agar, it is the bile salts and the crystal violet that's toxic to gram-positives. So therefore, we're selecting for the growth of gram-positives. So you'll notice on this top plate, you have E. coli. E. coli is a gram-negative bacteria. It very easily can grow in the presence of the crystal violet. That dye, while those cells are alive, can't get in past their outer membrane. The bile salts, again, doesn't affect them. It doesn't get in. It doesn't damage them. Unlike gram-positives, that dye will rush right in. Those bile salts will interfere with them and kill those cells. The bottom plate is Salmonella, which is also another gram-negative bacteria. And so we would use this type of media to grow them. This media then can go one step further. Once you've selected for your gram-negatives, you can ask an additional question about these gram-negatives. Of course, only the gram-negatives, because we did what to the gram-positives? We killed them, so we can't ask any more questions of them, right? They're dead. So our gram-negatives, on the other hand, though, there is lactose in this media. And most bacteria that can ferment sugars are going to produce what common byproducts? What do we like that we can make from fermentation of sugar? Alcohol is one. But another one that starts with an A that you can use a pH indicator to test for, acids, right? And gases. So here, acids, when they're produced, just simply having a pH indicator like neutral red in the media is going to help us detect this byproduct of sugar fermentation. So in the case of neutral red, it'll turn kind of pinkish in the presence of acids. So E. coli is a really strong lactose fermenter. Do you see the pink colorization here? We're going to notice how this one's nice and white. The Salmonella is not a lactose fermenter. E. coli, on the other hand, is. And we talked about him before, right? And his love for lactose. So if it's gram-negative with this media, since we are selecting for gram-negatives, we can also differentiate between the gram-negatives for lactose fermentation by having the lactose in the media along with a pH indicator, in this case neutral red. So we talked about blood agars. We said it is enriched with having those whole red blood cells, and it's differential and helps us differentiate the different hemolysins, beta versus alpha, or no hemolysis at all. All the rest of the medias, the McConkey and the two others that we're going to talk about, the eosin methylene blue and the mannitol salt, are all selective. So again, whenever we say selective, we're looking to see what is toxic in that media that's going to interfere with the growth of particular organisms. And then if it's differential, what are we asking? What question are we asking? Basically, what can they do, right? And the most common question we ask as far as differentiation, other than red blood cell destruction, is fermentation of sugars. So all of these, right, media ask about fermentation of sugar. The eosin methylene blue, the McConkey, as we've already said, it's about lactose fermentation. And mannitol salt agar, mannitol is the sugar alcohol that can be fermented by some of them. For eosin methylene blue, its name is telling you what's toxic. It has both eosin and methylene blue. Those are two dyes. And so again, we're killing off gram positives and selecting for the growth of gram negatives. And this media is very similar to McConkey, and that's why we use EMB in the microbiology lab and we don't use McConkey, because they basically do the same thing, just in a different way. Some formulas of eosin methylene blue can actually ask a little bit more information, whether they can do sucrose or sugar, straight up table sugar fermentation. The formula that we use in the microbiology lab only has lactose in it. So we're really only asking the question, can it ferment lactose? And this is one of my favorite ones, as you'll see in a moment. When it produces those acids, they interact with the dyes, which in this case the dyes are dual purpose. They're killing off the gram positives, and they're helping us determine the pH as they interact with the acids and the color changes that happen with that interaction. And especially for E. coli, which is a really strong lactose fermenter, produces a lot of acid, you get this really pretty metallic green sheen to the plates. So, you know, microbiology can be beautiful at times even. And these are some of my favorite ones because of that green sheen that they produce. The mannitol salt agar, as the name implies, other than the mannitol that gets fermented by some of the organisms, the salt is the inhibiting factor. And in this case, it's a high salt concentration. It's much higher than normal. So only salt-tolerant organisms will grow. So we're not talking gram-positive or gram-negative here with mannitol salt agar. We're talking about tolerance to salt. And there's a particular group of bacteria we're typically looking for when we use this media. We're looking for staphylococci, because staphylococci tend to be salt-tolerant. So they'll grow in the presence of this very high salt. And then one particular species of Staphylococcus aureus, which tends to be pathogenic, disease-causing, right, can ferment mannitol. And again, when they ferment sugars, what's a common byproduct we can very easily detect the presence of? Acids, right? So again, we've got a pH indicator in this media that helps us detect. In this case, it's phenol red, which is going to turn this really bright yellow, beautiful color in the presence of acid. So here's our EMBs. And as I said, the pictures don't do the real cultures justice, right? It's so much prettier in person.
Are these mediums considered complex or defined?
We'd have to look at their individual formulas to know that answer. So he asked, is it complex or defined. And I think most of these are complex. But I know there's like one in the microbiology lab that we use that ends up being defined, but I don't think it's one of these. So remember, what's the difference? What's complexed? What does that mean? Or on the flip side, what does defined or synthetic mean?
Synthetics man-made?
Yeah, somewhat man-made, right? The defined, we know exactly what's in there, right?
Yeah.
Where the complex, there just one thing, if we don't know what it is, like, you know, digest of animal tissue, y'all. What is that? How much protein is that? How much fat? How much? We don't know, right? And it's going to vary from batch to batch. But again, a lot of times that doesn't matter. We just need them to get the nutrients that they need to grow. So media doesn't necessarily need to be defined unless you need all those variables controlled. So although these medias are helping us select and differentiate, they're also probably classified as complex. If you're to look at their formulas, there's some components we don't know the exact concentrations. Excellent question. Thank you. But in order to ask you guys that question, I'd have to, you know, give you the what's in this. In microbiology lab, you guys actually have that in your lab manual. Whenever we use a media, it tells you exactly what's in the food we're giving our bacteria. Okay, so E. coli, as I mentioned, is gram-negative. So it's growing on this EMB plate, this eosin methylene blue. This plate looks kind of purple when you look at it because the combination of eosin, which is kind of a reddish dye, and methylene blue, which of course is blue, you get a purplish color, right? Red and blue make purple. And it is really the interaction of these dyes with the acid that E. coli is producing in the presence of lactose that it's going to create this really pretty metallic green sheen. This is not how E. coli normally looks, right? It's the interaction with the dyes in this media and the acid that E. coli is producing as it's fermenting lactose. Serratia mercescens, on the other hand, is another gram-negative, evident by the fact that it is growing on EMB. But you'll see that it almost even blends in with the plate color. It doesn't turn any of the pretty colors that you see when acid is produced. So this guy is not an acid producer, or not enough to cause the color changes here. So the other colors that you'll see, and I don't think I have any pictures in this one, no, are pink and purples as they interact with the acids, as the acids interact with the dyes in this media. So EMB is really cool in that we can actually kind of tell the level of acid production. It's not a yes or no, it's, oh yes, E. coli, a whole lot of acid. Other organisms, a little bit less acid production, but still fermenting the lactose. For mannitol salt agar, as I said, mostly in clinical specimens, right, from people, we're looking for typically staphylococci because these guys can grow in this high salt concentration environment. So this one happens to be Staphylococcus epididymis, and these plates are naturally pink because of the pH indicator phenol red in them. When an organism produces acids, it turns it yellow. So you can see here, this is a properly done streak plate, and this is a non-properly done streak plate, something we'll talk about in a moment. And so here you can see that the organisms are growing and are producing the acid, and it's turning it yellow. There's less organisms over here, so not as much acid has been produced, so it's the intermediate color, that kind of orange color. And as time goes on, as it produces more acid and more growth, the whole plate would turn yellow. And it's Staphylococcus aureus that can ferment this mannitol, this sugar alcohol, and produce this acid and yellow color change. So you know me, I love tables, right? Tables are a really good way of organizing information. So for blood agar, is it enriched? Yes, the adding of the whole red blood cells enriches that media. So it's a base media that they're actually adding stuff to it, right? So they're enriching it. Are there any selective agents in the blood agar that inhibit growth? Did we talk about inhibiting? No, we did not. Doesn't have it. Now this is different from in lab. In lab, when we use the blood agar plates that we use, have antibiotics added to them. So they are selective because of the antibiotics. But straight up what they're talking about in your book here is just straight up blood agar. So it is not selective at all. So it's not selecting or inhibiting any particular organisms from growing. Is it differential though? Yeah. Changes are going to happen. What in the media is going to be changed by the bacteria? What do we have to have in this media?
Red blood cells.
We got to have red blood cells. Is there a pH indicator in this media? No, we're not looking for any acid changes. What color changes or changes do we expect to see? What are we looking for when we use blood agar plates? We're looking for the lysis, right? Remember, there's two types of lysis. Alpha, which is incomplete, that greenish discolorization, and beta, which remember, bad, complete lysis of the red blood cells. So is A and B an enriched media? Eosin methylene blue? No. Does it have selective agents, though? Yeah, what are they?
Methylene blue.
The two dyes, right? Eosin and methylene blue. What organisms are inhibited by these dyes? Gram-positive. So therefore, who are we selecting for? Who's going to grow? Gram-negatives. What's the differential agent? Right? Can we differentiate some difference between our gram-negatives with this media? Yeah. And we're looking for sugar, lactose, and or sucrose. And then, as I said, in our lab, we use the formula that just contains lactose. We're just asking that one question. Is there a pH indicator in this media?
The two dyes.
Yeah, the two dyes act as the pH indicators. And what type of colors are we looking for here? And what does it tell us? It tells us lactose or sugar fermentation, right? And you're going to get metallic green sheen for those really strong fermenters, like E. coli. But you also see pinks and purples. McConkey agar, is that enriched? No. Does it have selective agents? Yes. What are they? Right, crystal violet and bile salts. Who does it inhibit? Gram-positives. So who's going to grow then, of course? Gram-negatives. What's our differential agent in this media? What are they going to change? They're going to, what sugar? This one's lactose only. Does this one have a pH indicator? Yes, it's neutral red. And so again, we're looking for color changes in this pH indicator that's going to let us know if acid was produced. So acid production is going to produce red growth. Does MSA, mannitol salt agar, have an enrichment?
No.
Nope. Is it selective?
High concentration of salt, 75%.
Yep. High salt concentration, right? 7.5%. It's really salty. So who is it going to inhibit? So if we've got a lot of salt, it's going to inhibit the ones that don't like salt, right? That aren't salt tolerant. So non-salt tolerant organisms will be affected. So who's going to grow? The ones that can tolerate that salt, right? And the most common ones we're looking for are staphylococci, are pretty salt tolerant group of bacteria. So for those staphylococci, can we differentiate any ability, and what ability? What do we have there that they're going to change? The mannitol, right, which is a sugar alcohol. Is there a pH indicator?
Phenol red.
Yep, phenol red. So this one turns what color in the presence of acid from fermentation of sugar? Yellow. And again, typically we're looking for Staphylococci aureus. Do you need to know this in this level of detail? Yeah, otherwise I wouldn't bother making a table. Sorry, y'all. Those of you guys in lab will love me later because you'll already know it, right? So, in lab, students actually do these techniques, and we'll see one of them demonstrated today. So you've already done streak plate. I'm going to demonstrate spread plate today and next week. And we don't do pour plate anymore because it's not easy. But all of these can help us obtain what's called a pure culture. So first of all, let's define what a pure culture or a pure colony and what a colony is. So a pure culture is a population of cells arising from a single cell or single species. So when a culture is pure, we're only dealing with one type of organism, right? Like Staphylococcus aureus or E. coli. And this is important too because if we're studying things like lactose fermentation and you don't have a pure culture, you don't know which organism is fermenting the lactose and which one's not. Or if they're both fermenting it. Okay. So when we do these studies, when we put them, say, on blood agar, we want just one organism so we can get one answer and it not be ambiguous. So a lot of times, though, you'll get a sample and it's not pure. And so therefore you're going to need to make it pure. And so streak plate or pour plate or spread plate techniques will help you isolate the organisms from each other, right, to be able to generate what we call a pure culture or a pure colony. A colony is a growth that's on a solid media, so typically on a plate media. And again, this grouping of cells that's so large that you can see it with the naked eye, it's only pure if it was started by a single cell or cells of the same species, which, you know, is not always easy to get them apart from each other, right, and get these pure cultures. Sometimes we have to do these techniques more than once. So a colony is a growth that you can see, a physical growth on a solid culture media, typically a plate, like a petri dish. And it's only pure, right, that growth that you're looking at, if it arose from a single cell or cells of the same species. So when you look at colonies on plates and stuff, there's no guarantee that that is in fact pure, unless you do additional testing. So if we have a mixture, right, if we're given a mixture of cells or our stock culture gets contaminated, we can use these different methods to help spread out the bacteria from one another. So notice they're all done on plates. This is going to give us the greatest surface area to hopefully spread them out so you can generate colonies that hopefully are of a single species and therefore pure. So they all involve spreading that mixture of cells on the surface of the agar so we could separate them out. So streak plate method typically divides the plate up into different sections. When scientists are first starting out, they'll even draw themselves a little grid or almost like a map on the bottom of their plate where their food is. And as I said, students that are in microbiology lab this semester have already done this. right? You guys have already done streak plate. And so I have some videos from the internet, and I think a video of me actually doing it that I also posted for you guys on our Canvas site. Because sometimes seeing these things in action helps you understand what's going on. But typically what we're doing is we're creating dilution by spreading the bacteria out on the surface of the plate in such a way that we're picking up less and less bacteria as we go. So when we start out, we're going to take from our original sample and we're going to put it on a small section of the plate. So you'll see here, this is where they started because you have the greatest amount of growth. But you can't see the bacteria at this point when you're adding them to this plate. You only see them now because it's been incubated and they've grown to large numbers. So that's why a lot of times, as I said, beginning scientists will draw themselves a grid. So they keep track of where they put the bacteria on the plate. So they'll add it, and you've got a lot of bacteria. So then they're going to flame their loop, which is their instrument they use to move the bacteria from one place to another. This completely sterilizes the loop by incinerating it in the flame. This removes all bacteria from the loop. So now when they go back to the plate, they're only picking up bacteria that are already on that plate. So they're going to pick up less bacteria than from the original sample. So you'll see that they'll then go, right, from that first section into the next section, and they're crossing over in this space here. So again, you'll notice that the growth is a lot less, right, because they're picking up less bacteria. Actually, it was this way. And then the third time, they're going to flame their loop again, remove all bacteria. So they're only picking up bacteria that they spread into the second section, into the third, and then they'll repeat, flame loop, and do it again. So now you see you have what we call isolated colonies. Colonies all by themselves, not touching other ones. And if they truly arose from a single cell or a single species, it would be a pure culture. As I said, depending, you would have to do additional tests to be sure that you've achieved separating out these organisms. You've generated pure colonies or pure cultures. Spread plate technique is what I'm going to demonstrate in lab today. So we're going to dilute a sample of bacteria. And then I'm going to, they're going to be in a liquid media. So I'm going to use a measuring device to put a set volume on the plate, because in addition to separating out the bacteria, you can also use this to help enumerate them, and that's going to be the main reason for why I'm going to spread bacteria out on a plate. I'm going to use a plastic, I'm going to use a glass. Plastic would have to be pre-sterilized. I got some of those, but I'm going to use the glass ones. We dip it in ethanol and flame it in the flame to sterilize the glass rod. The glass rod is bent in such a way it almost looks like a hockey stick. We affectionately refer to them as hockey sticks sometimes, or just a spreader. And we'll use that to spread the liquid containing the bacteria out on the plate, right? So we're spreading them out so they take up the whole surface of the plate. So then after we incubate, hopefully if we didn't have too many bacteria, then they'll separate from each other. And you can see this was probably definitely a mixed culture because you have two different colors. Organisms typically are going to be just one color under set conditions. So if this plate was, say, incubated at 37 degrees, one of them produced this yellow pigmentation and the other one is kind of a whitish. As you can see here, these colonies are kind of running into each other, right? We really aren't getting good separation. Where these are pretty well separated and the color is pretty uniform, so the chance is that may be a pure culture. But you would do additional testing to be sure that you have a single organism there. Single species. Pour plate method, as I said, we don't really use that much. Very similar to spread plate in that you're going to usually dilute your sample several times so that you don't have a very large number of cells, so that when they're on the plate, they're separated from each other. The tricky part about this is that the diluted samples are mixed with the liquid agar. The liquid agar, if it's too hot, it will kill the organisms. If it's too cool, of course you won't be able to pour it. It will be solidified. So it's really tricky getting it at that right temperature. And then you would pour it into sterile petri dishes. So a very common method that I'm going to demonstrate in lab today is serial dilution. When you dilute in a series, you can create much more diluted samples than if you took a sample and put it into a very large volume. But if you dilute one time and then dilute again and dilute again, we'll refer to in a series, you continue to dilute the sample. So notice the green dots here, there's a lot of them. Right, so we took a small sample size, 1 ml, and put it into 9 mils. We did what's called a 1 to 10 or a 10-fold dilution. So there's about 10 times less cells here. And then they did it again. And so we get 10 times less, 10 times less, 10 times less. So that we get so few bacteria that hopefully one of the samples that we plated onto a plate gives us nice separation and a decent number of bacteria to work with, or in the case of counting, a statistically accurate range to count within. So the good news for you guys that are in lecture class, that's all we're going to talk about as it relates to dilution. Those of you guys in lab, you will hate dilution, but I'm going to try to make you love it, right? Math is fun. Yeah, I know. It's so hard to convince people of that sometimes. It can be. The awesome thing about math, y'all, is you can achieve a correct answer. And the scary thing is you can get there different ways, right? The important thing is find the way that works for you and be consistent, right? My son's school right now, they're teaching the kids like all these different ways to do math. And some of us parents have never done any of these ways and we're like losing our minds.
Are you talking about that Common Core bullcrap?
It's not Common Core, y'all. Different ways of doing math has been around forever. Common Core, there's a misconception out there. Common Core is a set of standards of what objectives students should be able to achieve at grade level across the nation. It is probably the best thing we've ever came up with, and stupidity is trying to get rid of it. So that's all I'm going to say about that. If you look at it, the core is correct. It's how some people are approaching it that's difficult.
Not when they get graded because they didn't do it
Yeah,
that way.
right, yeah. That I don't agree with. And that's why I said that's how people are employing it, not the purpose of the system. So that's the unfortunate thing. The good thing about my son's teacher, I was like, okay, me and Dean don't understand this like arrow method. It don't make any sense to us. Can we do the place value or the other stuff? And his teacher just grades on whether they got it right or wrong and don't make them do specific strategies. And that's really the way it should be. You know, for math, the way I learned is if I had to do it the way other people do it, I'd probably get it wrong because it doesn't, like you said, make sense for some of us. You have to find the method that works for you and stick with it.
My daughter literally got an F because she wrote the times tables numbers backwards, like 9 times 2 instead of 2 times 9. She got an F on her math test. And on another
I would
school,
argue with that teacher in that school.
That's how all
BS!
That's all
It is.
public schools in Jefferson Parish.
Yeah. So that's where, you know, rigidity can be bad. But math can actually be really cool, right? It can be really cool. And it governs our lives, y'all. Look at the budget right now. Frightening. Okay. So, if math is frightening for you, it's not frightening for me. I even struggle through, you know, all these new ways of doing math with my son currently in 2nd grade. I used to tutor math all the way up to calculus. It makes sense to me. I can do it. And I can help you make sense of it. So don't shy away from me. Come see me for help. Right? And we'll get through dilution. Because if you're going to go and work in a lab somewhere someday, You will get fired if you can't do this. And I'm not kidding you. Okay? So, it's important. And it's okay to ask for help. I mean, when I was doing dilutions, when I was in research, I'd write out my plan and what I was going to do, and I'd ask one of my colleagues to check it for me, right? To make sure I didn't screw anything up, or I'd check it on the calculator and check it with my colleague to make sure I didn't plug numbers in wrong or something like that before I went and used all these reagents and potentially screwed up an experiment, right? So, that's what you should always do. So that's it for growth, the part by itself. Now we're going to move on to growth and control. So I'm actually going to